Hello everyone, I am Gungun Surana from India. Today I'm going to talk about the relevance of genes on survival of breast cancer patients. Breast cancer is one of the most common cancers affecting women globally. It can also occur in men. It originates in the cells of the breast, primarily in the ductal carcinomas or the lobular carcinomas. The risk factors of breast cancer include age, family history and genetic mutations, reproductive history, hormonal factors, and lifestyle factors. The 5-year relative survival rate for localized breast cancer is approximately 99%. For breast cancer that has spread to regional lymph nodes is approximately 86%, and metastatic breast cancer is approximately 29%. For my study, I utilized the Kaplan-Meier survival analysis and the univariate Cox proportional hazards analysis. In medical research, the Kaplan-Meier survival analysis is often used to measure the fraction of patients living for a certain amount of time after treatment. It constructs a survival curve that visually represents the proportion of subjects surviving over specified time intervals, providing valuable insights into factors influencing outcomes and treatment efficacy. The univariate Cox proportional hazards analysis is a statistical method to explore the relationship between survival time and one predictor variable, assuming the effect is constant over time. It has quite a few advantages and applications, including handling censored data, estimating effect of predictors on survival, identifying risk factors for diseases, and so on. Now, let me explain the approach I used in this study. I began by downloading and integrating the GSE 7390 dataset using GeoParse. From there, I extracted both merged expression and clinical data, paving the way to construct Kaplan-Meier survival curves based on covariates, followed by performing univariate Cox proportional hazards analysis for each gene. And finally, I extracted beta, hazard ratio, wall test, and p-value for each gene. Here are a few questions which I tried to answer during the course of the project. On what factors does survival of breast cancer patients depend? Which genes impact survival differences between the patients? What pathways are enriched among genes that significantly impact survival differences? Here are my results. Estrogen receptor presence is associated with better prognosis and survival. It is often associated with slower growing and less aggressive as compared to estrogen receptor absence. Estrogen receptors are basically proteins found in some breast cancer cells that bind to estrogen, which is a hormone that can promote the growth of breast cancer cells. This is a forest plot that summarizes the hazard ratio across the top 10 genes as shown. Gene CIA-PIN1 has the highest hazard ratio at 2.77. This gene is involved in cell growth and survival and is often reduced in expression in breast cancer. If hazard ratio is less than 1, then it often has a protective effect. The gene ARL6IP5 is often repressed in some breast cancers and acts as a tumor suppressor. This graph shows the top 10 enriched items in this study. Dysregulation of translation processes and ribosome assembly in the cytoplasm can lead to altered protein synthesis, promoting uncontrolled cell growth and cancer. These are the key takeaways. Disruptions in processes like cytoplasmic translation, cytosolic ribosome function, nucleoplasm integrity, and translation initiation can lead to altered protein synthesis, genome instability, and faulty signaling in breast cancer. These changes contribute to uncontrolled cell growth, tumorigenesis, and cancer progression. The survival analysis identified key genes associated with overall survival in the GSE 7390 dataset. These genes can potentially serve as biomarkers for breast cancer prognosis. Future work includes looking at these candidate genes' expression profiles and tumor subtyping analysis. Thank you.